controversy around so-called gain-of-function research has erupted alongside the heated debate around the coronavirus pandemic's origin. It is a type of research that makes pathogens like viruses deadlier or more infectious to humans. The researchers intentionally modify the virus in lab experiments to give it new functions. For instance, the ability to spread by air or attack human nerve cells. Gain-of-function research seeks to make a virus more easily transmissible between humans, to increase the severity of the disease it triggers, or to make it resistant to existing treatments or vaccines.
Two main reasons are given for why this type of research is done. One is to better predict which viruses in animal populations might one day naturally mutate and become infectious for humans. The second is to be better prepared when a new pandemic hits, for instance by already developing a vaccine for it preemptively.
So how many vaccines have been developed as a result of gain-of-function research?
None. There have been no preemptive vaccines to date.
Have any pandemics been prevented thanks to gain-of-function research?
The answer is not that I know of.
What's clear is that it didn't prevent this coronavirus pandemic.
Actually, none of the gain-of-function research that was done over the past decades or whatever, none of it contributed to finding a vaccine or cure for SARS-CoV-2, for COVID-19.
In the effort to protect us from a pandemic, scientists could cause a pandemic.
So let's look a bit more closely at how gain-of-function research is done. One method is to genetically re-engineer pathogens by inserting genes from one virus into another to make it more aggressive.
Another, simpler method is called animal passaging. Researchers inject a virus into ferrets or so-called humanized mice. These mice have been genetically manipulated to have human lung receptors. The virus undergoes slight mutations as it passes through several generations of mice. The scientists select the strongest mutations until the virus eventually becomes transmissible to humans.
You pass the virus through an animal host over and over again. So you're basically speeding up nature. You're like speeding up evolution by hundreds or thousands of years.
Making the disease the virus triggers more deadly is one possible aim of gain-of-function research. Another is to make a virus that would normally only attack animals better able to infect a different type of host, humans. In 2011, an extreme gain-of-function experiment rocked the science world. Scientists used the serial passaging method to make the H5N1 avian flu virus more transmissible to humans. The lead scientist, Ron Fouchier, claimed he created one of the most dangerous viruses you can make.
They had taken a virus that was deadly but not good at infecting humans and made it also very infectious. So that kind of research, I think, is the risks are immensely high. If it broke out, there would be just widespread death. But the benefits, what are they? Many scientists look at this experiment and they think, why did you do this?
The thing with the avian flu is that it is extremely deadly, with an infection fatality rate of approximately 60%. Compare that to a rate of under 1% for COVID-19.
A coronavirus found in bats in a remote Chinese copper mine kills 3 miners in 2012. The abandoned mine is near the tiny town of Tugun, deep in China's southwest Yunnan province. Unknown to the men working there, the bats populating the mine and the bat feces they were shoveling contained an unknown coronavirus that would later be named RATG13. Around 2 weeks after entering the mine, the miners fell sick with a strange flu-like illness. Their illness eerily similar to COVID-19. The virus samples are studied in the biohazard laboratory in the city where the pandemic first broke, Wuhan. These Chinese medical records, kept secret for years, showed their symptoms were identical to COVID-19. And samples of the bat coronavirus taken from the feces in the mine were also taken to the Wuhan lab. Comparison tests would later reveal it was a 96% match to COVID-19. The Wuhan lab has been conducting highly risky research designed to genetically engineer highly lethal human viruses in order to study them. So, Could COVID-19 have accidentally leaked from this laboratory?
The very simple data elements on the ground all point to the likelihood that this virus came from a lab.
As the world seeks answers about the origins of the virus, what was once dismissed as a conspiracy theory has become a legitimate scientific debate. Is it possible that COVID-19 already existed and was being researched and accidentally escaped from the Wuhan lab, sparking the pandemic? But it's a debate many scientists reject. There is evidence that the Wuhan Institute of Virology was carrying out research on the bat coronavirus called RATG13 and on samples from the dead miners. At the time, the bat virus, RATG13, was found in the copper mine, the most secure laboratory in China for the study and containment of viruses was the Wuhan Institute of Virology. It was and still is run by Shi Zhangli, who has confirmed that she did take samples of the copper mine virus to study in Wuhan. It involved the manipulation of viruses for research, which made them incredibly dangerous. The bat virus that infected the miners in the copper mine could have been deliberately mutated into COVID-19 through the controversial and risky research technique called gain-of-function engineering. Shi Zhangli has confirmed that she and her team conducted gain-of-function engineering on bat coronaviruses at the Wuhan Institute of Virology.
Researchers at the Wuhan Institute of Virology were collecting and researching bat coronaviruses and using humanized mice prior to the COVID-19 outbreak. High-security BSL-4 labs built for research on highly infectious pathogens are booming around the world, and 3/4 of them are located in densely populated urban areas. But surprisingly, gain-of-function experiments are not done exclusively in highest-security BSL-4 labs. Some are done in level 2 and 3 labs.
We recently found out that the Wuhan Institute of Virology was doing a lot of this SARS-like virus research at a very low biosafety level.
The Wuhan Institute of Virology was actively working with SARS-CoV-2 viruses over many years, including the closest known relative to the pandemic virus, using inadequate safety protocols. They also discovered that the lab and Chinese authorities have methodically attempted to conceal these activities.
Viral pneumonia has hit central China's Wuhan city. Authorities have reported 27 cases in total, 7 of which are critical. Patients are reported to have worked at a local seafood market.
What is puzzling for scientists is it's now known that many of the first COVID-19 victims were not connected to the market in Wuhan, where it was first thought the virus emerged.
The first 2 cases hadn't been to the wet market from the Wuhan cluster, and half of the cases in that cluster had not been to the wet market.
And despite extensive testing, no trace of the COVID-19 virus was found in the animals that were sold at the market.
They went to the wet market, they took samples, they took environmental samples, but none of those samples showed a coronavirus in the animals that were sampled. So at Huanan Seafood Market, there's no animal samples that we know of that have a SARS coronavirus that could be an ancestor to what we see today and that has infected the world, essentially.
武汉市公安局官方微博发布消息称，关于当前武汉市肺炎疫情绝不姑息，希望广大网民遵守相关法律法规，不造谣、不信谣、不伪传。
The laboratory leak is a very reasonable hypothesis because it's happened before throughout history and in many countries.
There are legitimate questions the world still needs answers to about the origins of the virus. And that includes the role, if any, played by the bat virus that killed 3 copper miners 8 years before the pandemic. Legitimate questions, too, about the research carried out by the Wuhan Institute of Virology and its use of controversial genetic engineering that can create deadly viruses inside a lab.